thank you everyone for being here behind of your screen and listening to these talks. And of course, thank you for the organizer for organizing everything despite all the difficulties and giving us this opportunity to discuss. So what I'm going to present today is work that I did with my postdoc advisors, Philippe Lemay and Marc Suchard. So Philippe Lemay is at KU Leuven in Belgium in a virology lab, and Marc Suchard is a statistician in UCLA, so in California. And this is joint work also with Lam Ho, who is now in Canada, and Guy Bell, who is also in KU Leuven. And I'm going to talk about a Bayesian framework to study viral trait evolution on a tree. So the main framework of this talk is phylogenetic comparative methods. So this term comes from the ecology field, and the basic idea is that you have some continuous traits that you measure on a population of species, and you want to compare them between another. So for instance, here you would look at the New World monkeys, so many kind of small monkeys, and for each of them you would measure some continuous traits. So here, for instance, traits associated with the shape of the skull of these different species of monkeys. And what you want to do is to say something about the distribution of these traits among all the species of monkeys. And something that you need to take into account is that all those species are not independent from one another. So you cannot apply standard statistical methods out of the box, because you need to take into account the fact that all these species are somehow correlated because they share an evolutionary history. And this history is here represented by this phylogenetic tree here. So it's 3 times the same phylogenetic tree. And here, so the root of the tree would be the common ancestor of all the New World monkeys in the world. So it would live about 50 million years ago. So here I assume that the tree is calibrated in time. And each split event in the tree is a speciation event, so one species giving rise to 2 species. And then at the tip of the tree, you would have extant species that you can actually measure. So this is the origin for the term phylogenetic comparative methods. And it's been extensively studied for this kind of organisms. But actually, this kind of problem where you have continuous measurements at the tip of a tree can also be seen in another field, which is virology. So here I have 2 examples. So this one is the rabies virus in a skunk in the US. So it's not really visible, but the idea is that here all the dots are viruses that you manage to catch and sequence. And then they are linked by some kind of tree. So the tree is not very visible because it's on a map. So here, what you would actually measure is the position of the sequence that you manage to catch. So latitude and longitude. And so that would be the measurement at the tip of the tree. And the tree would be linking all these sequences together because the virus is evolving at the same time as it is spreading on the map. So here, the tree is not very much visible. So I have another example here. So this is a flu example. And the trait measured is some kind of antigenic measurement. And here, why do I choose this tree? Because we can see that it's very different from this tree here. So it's still a tree. Measurements still happen at the tip of the tree. But here, this tree was spanning over 50 million years, while this one is only about 10 years. So from around 2000 to 2007. So that is because viruses evolve much faster than monkeys, for instance. And so what it also means is that we have tips dynamically in time. So we can sample this process at different time points. On this example, if I measure species of monkey now or 10 years ago, it doesn't make any difference because the tree is very big. But here, it does make a difference, and we can exploit this temporal signal to say something about the distribution of the tree. So the question we want to ask is, what is the trait dynamic for an organism that is evolving at the same time as its trait is also evolving? And as you can see, there is a wide range of possible applications. So we want something that is flexible enough to be able to take into account these various configurations. So the basic plan for this talk is, first I'm going to talk about models of trait evolution. So what kind of model can we apply on these phylogenetic trees? And then how can we do this, the inference for these models? And finally, I will present an example for the study of the heritability of HIV. So do not hesitate to ask questions if something is not clear. I can always answer, and if this introduction is okay, I will move on to the models. So what kind of model are we going to study? So here, so this is a basic model we're going to use. And the idea is that for now, I'm assuming that the phylogenetic tree is known and is calibrated in time. So I'm going to talk later about this assumption, but for this model, I'm assuming that the tree is known. And here, I have some measurements y1 to y5. So these are continuous traits, and I measure them at different time points. So here, this is a dummy example from 2006 to 2008. And what I'm going to do is to model the evolution of the phenotype of the continuous trait as a stochastic process. And here, the simplest stochastic process possible would be the Brownian motion, BM. And this is some kind of branching process, but kind of degenerated one, because we know for sure the branching times here. So what is happening here? So this ancestral red species here, its trait is evolving according to a Brownian motion. So it's represented here. And when there is a speciation event, so the red species giving birth to the two orange species here, then the stochastic process is split into two independent stochastic processes that start at the same point and that have the same distribution. So here you can see the two shades of oranges. And then for this speciation event here, you have again a split. And then at the tip of the tree, you stop the process and you actually observe it. So in this model, we never actually see these trajectories because we don't have access to this part of the tree, because this is the path that you didn't sample. All you have access to is the last value of the process for each of the tips. Okay, but doing that, you have the kind of structure that you want on your observations, because you can express the covariance between two tips. So for instance, here, y4 and y5. And this covariance will be proportional to the time of shared evolution here. So noted V45. So this is the time between the root of the tree and the most recent common ancestor between the two tips, y5 and y4. And this is just sigma square time this time here, and sigma square would be the variance of the Brownian motion that you took. And this is the kind of structure that you actually want, because what does this mean? This means that if two species are closely related, so if they are close sisters, then their time of shared evolution will be long, and so they will be highly correlated. So what you're saying is that two species that have evolved, that are very close in the tree, should have traits that look alike a lot. While two species that are far away in the tree, so their time of shared evolution will be small, and so we expect that they do not look that much alike. Okay, so we have the covariance. If we have just simple Brownian motion, then the expectation doesn't change. It's just the ancestral value of the expectation at the root. So here it would be, for instance, 0. So you can express this as just a normal vector. So y will be the vector of observation 1 y1 to y5. So each of them have the same mean mu here, and they have a variance that is given by sigma square times v, where v is this matrix of shared times of evolution. And if the tree is known, so if I assume that the tree is known, then v is known. So this is a very standard distribution. And if I don't like v, I can do a Cholesky and I can get rid of this known matrix. So here, this is just for one measurement, but actually, usually you don't have only one measurement, you have possibly many measurements for each of the tips. So here, this is just a simulated example to show that I could have like 2 traits, for instance, for each of the tips. And so instead of measuring only one trait for each of the tip I would measure p traits for each of the tip. And so I just need to take multivariate Brownian motion, where the variance of this Brownian motion, so the variance matrix is denoted R, so R for rate matrix. And the covariance between 2 tips is very easy. So the covariance between trait K of tip I and trait L of tip J, this is just proportional to the time of shared evolution between the 2 tips I and J, so Vij, times the covariance between traits K and L. Okay, so this is a very natural structure to have. It's just very similar to this structure we had here, except that it is multivariate. Okay, and the variance, same than before, it doesn't change. So this covariance structure can be written like that. So this is a matrix normal distribution. So Y here is a matrix with n rows that are the species and p columns that are the p traits. And so this is a matrix normal with mean mu, with variance for the rows given by v. So this known matrix if the tree is known, and with column variance given by R, so the variance of the Brownian motion process that you took. So this matrix normal, it just means that if you take the big vector of the vectorized version of this matrix Y, so if you take all the columns and you stack them on top of each other, then the variance of this thing is just a Kronecker product between R and V. So what we see here is that the contribution of the tree given by V and the contribution of the process to the variance are very well separated. It's just a Kronecker product. So this means that for the inference, it's going to make things really easy because of this Kronecker product that we have here. But I'm going to come back to that in a moment. So here, I took the most simple possible stochastic process, which is a Brownian motion. But the Brownian motion is just basically noise, so it would be just random drift. And the variance is just linearly raising when the time augments, because it's proportional to the total time. I'm stuck. Okay, so Brownian motion is the simplest process I can imagine. So the next simplest process I can imagine is the Orchten-Uhrenbeck process. So the idea is that you still have this Brownian stochastic part here, but you add this recall term. So beta would be interpreted as some kind of optimal value for the trait in a given environment. And then your process is going to fluctuate according to a Brownian, but its mean is going to go to this optimal value here, beta, with a speed that is controlled by alpha, so the recall parameters or the selection strength parameter. Okay, so the bigger the alpha, the faster you go to your optima. And you go there with some kind of exponential decay in expectation here. And what you can look at for interpretation reasons is the time, the phylogenetic half-life. So this is the time between to go from where you are to half of where you go. So this is this time here. So log of 2 over alpha. And this time you can compare to the total height of the tree. So if this time is very short compared to the total time in your tree, then it means that you have enough time to go to your optimum and you're gonna have some hopes of seeing this optimum. While if on the contrary, this time of shared evolution is very big compared to the tree, it means that in your evolutionary time, you don't have time to go to your optimum, so you won't see your optimum. So it gives a natural scale to interpret this alpha parameter. Okay, so this is very similar to a Brownian, except you have this deterministic term in addition to it. So if you put it on a tree, it's the same thing. So it's the same mechanic. Each time you have a speciation event, you split into two independent OE processes with the same parameters. And here the optimal value is 0, so everyone sticks around 0 more or less. And you can express the moments of the observation and the chips. So basically what you can see here is the covariance. You have an explicit form, and what you can see is that it still depends on the time of shared evolution. And what's kind of important is that it's bounded by sigma square over 2 alpha. So while the Brownian motion had a variance that could explode linearly in time, here it's bounded by sigma square over 2 alpha, and this is precisely the variance of the stationary solution of the Ornstein-Uhlenbeck process. And if you look at the expectation, so here, basically you have some kind of actualization term because you're going from where you started to the optimum. So if you leave enough time, you're going to be close to the optimum. And if you start early, then you're going to be close to the ancestral value. So again, this is for just univariate traits, and maybe we want multivariate traits. So here, it's kind of the same thing as for the Brownian motion, except that this term here becomes a little bit more tricky because here, so instead of one beta value, you have a vector of beta values, of course. And here, instead of one parameter alpha for the selection strength, you have a matrix controlling the whole dynamic of your traits going to your optimum. So depending on the shape of this matrix A, you can have different kind of behavior of this Ornstein-Uhlenbeck process. So the simplest form you can imagine would be A would be scalar, so diagonal with the same value on the diagonal. And what I'm representing here, so here I'm representing the vector fields for just the deterministic part of the equation here. So I'm forgetting about the Brownian part here. This is just the deterministic part. And what I'm doing is that I start the process here at 0, and I have an optimum which is half and half here. And so here, for this kind of matrix, all the vector fields is attracted directly to the optimum, and I go there in a straight line. And what I represented here is still only the deterministic part of the equation. And I made this thing evolve on a very specific tree, so some kind of ladder tree, where I sample the process somehow dynamically in time on a very nice ladder. And this allows me to show you that the trait is evolving. So from 0 to 1/2, the further you are from the root. Okay, so here both traits go to 1/2 with the same speed. So of course I could take the same thing but diagonal instead of the same value here. So here the selection strength is a bit stronger on Y. So the process first go to Y and then to X like this. And you can see it here where the process in Y goes faster than the process in X. The next step is that you could take A symmetric. So here you're diagonalizable in an orthonormal basis. So basically the same thing, but you just turn your head. And what it means is that the only difference is that you can maybe go over your optimum before going back here. So just because you turn your axe. So here basically for Y, you could go over 1/2 and then go back to 1/2. So that's the kind of behavior you can expect with this kind of matrix. And to go one step further, you can imagine that it's diagonalizable in R and with strictly positive eigenvalues. And here, so the axes are no longer orthogonal. So you can have weird things like this, where you go away from your optimum before going back to the optimal value. And so here, for instance, if I look at X, it first becomes negative and then goes towards 1/2. And then same for Y, it goes very high above 1/2 and then tries to go back to 1/2. So in general, you could have A diagonalizable in C with strictly positive real parts of the eigenvalues. So here you would start spinning around, but we are not going to consider this case here. Okay, so this is just to show that this Arstein-Wende process is already quite flexible model, and you could imagine modeling different kind of processes with this tool. Okay, so the assumption I'm making here is that it's on this form here with strictly positive eigenvalues. And if I do this assumption, I can write all the moments for y. There is a closed form expression, but instead of some kind of Kronecker product I used to have, I have now an Hadamard product, so term-a-term matrix product. And this is way less nice for the inference, we will see later, just after that. So it's still a big Gaussian vector, so this is nice, but we don't have this nice Kronecker decomposition, so it's not going to be as nice as for the Brownian motion. Okay, so now the goal is to do the inference of these two processes when they evolve on tree. So if you have any question on the models, maybe if anything is not clear, you can ask your question now, and otherwise I'm going to move to the inference of this process. Okay. So for the inference, so I'm going to do to try to do Bayesian inference. So what I'm actually measuring, so is y, the matrix of continuous trait observations, and in general, I don't actually see the phylogenetic tree because this is something that I construct, and I construct this from usually the sequences that I have. So this is a matrix of sequences for each of the tip of the tree. And what do I want to learn from these two matrices of observation? I want to learn theta, the parameters of the Brownian motion or Arstein-Wende giving rise to y. I want to learn the tree itself and some parameters associated with the tree. And as usual, so I decompose the posterior as the likelihood and the prior. And the fundamental assumption that I'm going to make here is that conditionally on the tree here, the process of the continuous trait and the process of the sequences are independent. So conditionally on the tree, y and s are assumed to be independent, so that I can factorize here this joint density into these two densities here. So one in y and one in s. And so basically this term here, so learning the tree from the sequences, is a classical problem in phylogeny, and there are already a lot of tools to deal with this problem here. So I'm not going to talk about this at all. What I'm interested in here is to learn this part here, so learning theta from the continuous observation. And so this is why I can assume that the tree is known, because on each step of my MCMC, I can assume that the tree is known, and then using all the other terms here, I can also update the tree in each step of the MCMC. So for one step of the MCMC, I can assume that the tree is known, and then the tree itself is updated during my big MCMC, taking into account all these terms here. So this reduces our problem for now to just this term here, assuming that the tree is known, is fixed. Okay, so for the Brownian motion, actually everything is kind of easy, because what do you want to do? You want to sample from the posterior. So what are the parameters? Just the variance matrix R and the mean matrix mu, and given your observation. And here the likelihood has a very simple form because it's a matrix normal. So it's something that is very well studied and known in the literature. So what you can show is that you can use conjugate priors. So if you take an inverse Wishart prior for your variance matrix, and then a normal prior conditionally for your expectation vector, then you can do a Gibbs sampler, and because you can express the distribution of one parameter given all the others, it's still an inverse Wishart matrix with some parameters that you can compute. And you have an automatic sampling of the space of variance matrix and also expectations, of course. And it's a Gibbs sampler, so you don't have a rejection. And basically, this is a very well-known problem. The only difference is that you have this V matrix here, but since you are assuming that the tree is known, this V is known, so it doesn't add any difficulty. So for this problem, you're happy. Every time you see a plus on the slide, it means that you have some explanatory slides. So if you have a question, please do not hesitate to ask. So here, for instance, if you want explanations on Gibbs, there would be some slides. So the problem with the Orstein-Uhlenbeck is that you don't have this nice form for the distribution. So you can't write conjugate priors. At least I can't write conjugate priors. Maybe you could, but couldn't find any. So you can't do Gibbs. And so you have to go back to a standard metropolis hashing, so a random walk. But you need this random walk to take place in a constrained space. So because you have two matrices here to sample from. And so if you were to sample each of the coefficients of the matrix independently, then you would step out of your constrained space very fast. So you wouldn't have variance covariance matrices that are symmetrics and definite positive here if you were to do that. So what you can show is that to do that, the only thing that you need is to find a transformation that is an homeomorphism mapping the space of constrained parameters to a space of unconstrained parameters, where Q will be the real dimension of your constrained space. So for instance, for a symmetric definite positive matrix, it would be something like n plus 1 over 2, something like that. Okay, so if you have a transformation, then you can just work in the unconstrained space, and then you have tricks to go back to the constrained space using the Jacobian of this transformation. Okay, and so what we can show is that we do have these transformations for all these parameters. So for the variance matrix, what we do is we write them in a correlation variance decomposition fashion. So here sigma would be the diagonal matrix with the standard errors, and then C would be the correlation matrix. So you really separate the scale effect, so the variance, from the intrinsic correlation effect. And so sigma is just positive parameters, so this is easy to sample from. And C is a correlation matrix, so it has strong constraints. And one transformation that you can use is called the LKJ transformation for the three authors, Lewandowski, I never remember, and Joe. And this dates back from 2009. So I was assuming that this kind of transformation existed from, I don't know, the 50s, but actually people have been interested in them relatively recently. So if you want more details on this transformation, I have some slides if you're interested now or later. And for the A matrix, so the selection strength matrix, well, assuming that is you have this kind of decomposition, then P are just unitary eigenvectors, and this is easy to sample from since they live on a sphere. And all these other parameters are just positive, so and possibly ordered for this lambda to ensure identifiability, and these are easy to sample from. And this transformation here also gives you a nice prior distribution for the correlation matrix. So for the Gibbs sampler, you do not have the choice for the prior. You have to take an inverse Wisher. Here you are much more flexible because you can have a prior for the scale matrix, so the variance matrix, and an independent prior for the intrinsic correlation matrix. And in particular, if you, so there is a parameter here, and if you take it to 1, then you'd have a uniform prior on all the correlation matrices. And you can also take it so that it looks more like an identity or more like very much correlated. So this gives you a lot of flexibility on the priors, this framework here. And so you have a running random walk MCMC, and you can do everything you want. And the question here, all my data is on a tree. And the question, can I use this tree to do efficient computations? Because if I do naive computations, then I will have some big matrices to invert. And what I can show here is that using the tree, you don't have to do that. And so to see that, I'm going to see the Brownian and the Ornstein-Uhlenbeck as instances of a general Gaussian propagation model. So the idea is that you have this kind of hierarchical model where you model the root as some kind of Gaussian, and then the distribution of one node, given its parent, will be also a Gaussian with a variance that depends on the branch. It can be a different variance for each of the branches. And then its expectation is linear in the value of its parent here. So for the Brownian motion, for instance, if you let the Brownian motion evolve for a given time t, then the result will be distributed according to a Gaussian centered on where you started and with a variance which is proportional to the time of evolution t. So that's exactly what you have here. So Q would be 1, R would be 0, and sigma j here would be Lj, so the length of the branch j, and R the variance of the Brownian motion process. You can write the same thing for the Ornstein-Uhlenbeck, basically the same thing, except that you have this actualization parameter here. And this model is very flexible because you could have many other processes that you can write in this way. For instance, the integrated OU, where you assume that the speed is following an OU, but the position is the integration of this. You could have some shifts also in the parameters, because here you have parameters, for branch parameters. So for instance, if you want to assume that in some part of the tree, the process is evolving faster than another part, then you could use this same framework. And also, because this is just a hierarchical Gaussian, you can add a layer here at the end and saying that instead of measuring the actual value of the process, you measure some kind of degenerate version of it, because you have maybe measurement error, or in the case of a species, well, you actually never see the actual ideal value of the species, but you see many individuals in this species. So this S matrix could also be some kind of intra-specific variation. And this you can add very easily because of this hierarchical structure here. Okay, and so as long as you have this very flexible framework, then what you can show is that you can compute the likelihood of the data at the tips linearly in the number of tips. So the naive algorithm would need to invert a matrix of size n times n, so basically n cube, n square, n cube. Here you are linear in n, so if you have a big tree, then it goes faster. And the idea is that you just do one post-order traversal of the tree, so from the tips to the root. And this is basically very similar to a Kalman filter, except that everything is on a tree, but the information propagation goes similarly. And it has many names in the literature because it has been invented over and over again many times. So here are some sample names. And the difficulty here is really numerical robustness, because when you have a big tree, then you need to propagate the information very deep in the tree. And if you have any error, it can lead to catastrophic numerical failures. So one big part of the work here was dealing with this numerical robustness. Okay, and what's even better is that once you are here, so you do one traversal from the tips to the root, and once you are at the root, you can do another trip back from the root to the tips, and this allows you to compute the gradient of the likelihood with respect to any branch parameter theta k. So any branch parameter here, you can compute its gradient also in a linear time in the number of tips. And this is just an application of Fisher identity, it's sometimes called. So you just write this gradient as an expectation given the tips. So that's why you need to go back here, because you have integrated all the information when you are at the root, and then you propagate it back to all the nodes here. And so once you can do that, it means that you can compute the gradient with respect to any parameter. And having the gradient is really something desirable when you want to do inference. You can use it in many ways to make your MCMC faster. One of the ways that we use is, so you can compute this in linear time. And one of the ways you can use it is to do Hamiltonian Monte Carlo. So this is basically a way to use this gradient information to sample faster from your posterior. But you could imagine any other kind of sampling methods that use the gradient, it will also work. And so here, everything is implemented in the BEAST software package. So BEAST 1.10. And this is useful because in BEAST, you already have all the machinery to do the inference of the tree given the sequence. So everything is already here. And here we just added a module to do a continuous straight inference using the tools I just presented. And so this allows us to do one big MCMC to infer at the same time the tree and the continuous process. And so this makes this thing quite flexible. And you can benefit from everything that is already in BEAST. So for instance, factor model or marginal likelihood estimation to do model selection and many things. Okay, so now I'm going to present a small example. I don't know if I have maybe 5 minutes left or a bit more. Or...
Yes, I think 5 or 6 minutes is okay.
Okay. So what I want to do is to estimate the heritability of the virulence of the HIV. So basically, why am I studying the virulence? Because intuitively, you can imagine that the virulence would follow an Orsten-Wenbeck process, because if you are too virulent, then you're going to kill your host very fast, and so you won't have time to propagate. And on the contrary, if you're not virulent enough, then you're going to be destroyed by the immunitary system, and you're not going to be able to propagate. So you'd have some kind of trade-off between too much virulence and not enough. So that's why we used this example for this intuitive notion. And how do we measure virulence? So you have two scores. One is a T-cell decline rate. So this is linked with virulence. And one is a set point viral load. So the idea is that when you're infected with HIV, first your viral load, so the number of viral entity in your body, increase sharply. And then your immunitary system kicks in and you can control the viral load to some kind of a plateau here. And this plateau can last for many years. And then if you're not cured, if you don't have any treatment, the virus wins and the viral load explodes again. And the actual value of this plateau is quite variable between individuals. And it's been shown as a good predictor for virulence of this virus. And so the question we want to ask is the virulence heritable or not? And so, of course, the virulence is some kind of an interaction between the virus and the host. But is it true that some strains are more virulent than others? Or is it just a question of luck? If I have one strain and one host, maybe the strain will be very virulent. But if it's another host, then it won't be virulent at all. So the question is this virulence heritable or not? So to measure that, we use our model here. And basically, the heritability is the ratio between the variance Z due to the stochastic process on the tree and the total variance, where you also have this S error variance, which is modeling all the interactions between host and virus that you do not take into account in your stochastic process. So if the heritability is 1, it means that the trait is perfectly heritable. So this S matrix will be basically 0. And if the heritability is 0, this S matrix will be much higher than the process matrix. And so basically you're saying that all that matters is the interaction between the host and the virus. So to fit this model, we are going to use 3 different models. So first, the Brownian motion for the 2 traits with correlation, then the Orsten-Wenbeck for the 2 traits with correlation. And one last model we want to use is the OU-BM, so OU for the viral load and Brownian motion for the T cell decline with correlations. And this is our favorite model. Given the literature on the subject, we would expect to find this model to be the best one. And we add an error, correlated error term on top of it. And we fit 8 of them using our HMC methodology. Here, the tree is fixed for this problem. And then we estimate the marginal likelihood for each of these models to select for the one we like the best. And here is the result. So you have either independent error or correlated error. So this is just, is S here diagonal or not? And then you have BM, OU, BM, and OU. And basically what you're saying, and this is the estimated marginal likelihood. So the higher, the better. And so here you would select for the independent error Orsten-Uhrenbeck. And this was kind of surprising for us because we would have expected this model to be selected. And so if you do select this model, you can look at the heritability estimates, and they are somewhat in line with the literature, although a little bit higher than what you would expect, and with kind of wide confidence credibility intervals here. And this result was really surprising. So we did a simulation study to kind of check them. And I think I'm short of time here, but the idea is that we took the exact same tree and we simulated according to OU-BM process, and then we added some noise. So when the factor is 1, it means that you have the same amount of noise as the variance of the process. So basically the heritability is around 50%. And so when the heritability is lower than 50%, you are on this range here. And when you are here, it means that you have a very small amount of error, so a very high heritability. And so what we observed here for this kind of heritability we see, it means that we are around this regime here. And for this regime, what happens? We can see that we are not selecting the right model in about 1/4 of the time. 20% of the time, we do not select the right model. So it means that the model is really noisy and it's hard to do model selection. And if you look at the heritability estimate, when we can see that they are usually overestimated and with a wide confidence interval. And so that's not really surprising in a way, because what you're saying is when you have 30% heritability, it means that in all your data, the variance, in all the variance of your data, the tree is only responsible for 30% of it. And within this 30%, you're trying to distinguish between OU or Brownian. And basically, this is just not enough data to really distinguish between the two very subtle differences between OU and Brownians that they live on the variance matrix. And so that's what the simulation basically show. So it means that you need to be really cautious when you estimate heritability for this kind of problem, because basically the power is very low. So in conclusion, we have a general framework for trait evolution with data tips. So here I presented an example in virology, but you could also imagine examples in ecology where you'd have some fossils and you'd have, again, data tips. So we have another example with the family of weasels. So if anyone were to ask me a question about weasel, I have some slides, and there is a picture of a skunk and a raccoon eating cat food in a Hollywood backyard. So if you were to ask a question, you would see this picture. And we showed, so we had efficient algorithm, flexible models and implementation. And in perspective, well, as I didn't mention it, but the identifiability of the full OU model has raised some questions. We only fitted diagonal OU models because we tried fitting more complex one, and it seems that we never have enough signals. So maybe there is some kind of theoretical identifiability issues that we couldn't see, or maybe it's just that with this kind of data, we never have enough signal to infer the full selection strength matrix. Of course, you could imagine shifting parameters. So for now, we assume that all the parameters are homogeneous across the whole tree, but of course for a big tree it's not realistic. So you could imagine ways to allow this parameter to shift with adapted priors on this shift. And of course you could apply that to different kind of questions. So geographical spread, for instance, and maybe more adapted model for that, or any other related questions. And so there is an archive paper for this, and it's in print in the Annals of Applied Statistics. And thank you very much for your attention. And if there is time for questions, I'm happy to answer them.
Thank you very much, Paul. Are there some questions? I'm just checking the chat if there is something. If not, I can start with a first question. Just to say, so we planned around 5 minutes of specific questions to the talk now, and for more general questions and interactions between the talks, it will be dedicated to the discussion session. So I ask you my question if nobody starts. When you spoke about the Orstein-Uhlenbeck and the possibility of making the Metropolis Hastings algorithm, so it is in a constrained space, you say, and at some point you have to unfold somehow the constraint function to map it to RD. I just wanted to know under which conditions is it possible, and does it have some biological interpretation? So do the conditions have a biological interpretation? In which settings we can unfold?
So yes, but basically, so for the Orstein-Uhlenbeck, you have two matrices to sample from. So the variance matrix, well, it is a variance matrix, so it has to be symmetric, positive, definite.
Mm-hmm.
So I don't know if it's a biological interpretation, but it has to be a variance matrix. So you can't. And then you have some standard transformation from this variance matrix space to the to a non-constrained space. And then for the A matrix, so if you make the general assumption that you can it's diagonalizable in and with positive eigenvalues. So biologically, I don't know exactly what it means. I guess it depends on the scale of what you're looking at, but it's a bit like what I showed when I showed the OU process. You'd have this kind of trajectories here for the most general matrix.
At least it means that the fixed point is stable, I guess?
Yes. So that's an assumption we make from the start, is that all the eigenvalues are strictly positive. Mm-hmm. But this is not really related with the Metropolis Hastings algorithm, because it would be very easy to sample from real eigenvalues, I guess. So this is really we want the optimum to be really an attractor of the process and not a repulsor.
So in fact, you have a wide range of models for which it's possible to.
Yeah, all these models, you could in theory fit them. At least all the machinery is here. So what I said is that actually, if you try to fit this on tree, the MCMC chain is not happy, and it's usually oscillating. It's very hard to converge, and even for we made some tries with very degenerated trees like that, and even for this kind of trees, that is why we stepped back, and all the application use actually a diagonal matrix, because it seems that we don't have usually enough signal for the kind of data we are looking at to infer this general process here. But maybe it's an identifiability issue that we didn't see, maybe some kind of interaction between full A and full sigma here. Maybe it's because of this data structure, this I can't really answer for now. I think you're frozen. Is it just me or?
Apparently, Lauren is crashed.
So maybe I can go on and answer Christophe's question, or maybe you.